So the second session is identifying the impurities and developing mitigation strategies. I think very important topic is this in industry, and now everyone is looking for this. So let me start. If this is the chromatogram, then formulation scientists always expectations purity 99.999%, total impurity is less than 0.001%. I know audience, analytical person, they never accept this kind of chromatogram because they knows realistic what happens in day to day. But yes, if this is the chromatogram, formulation scientist expecting and really they are happy for this. And not for that formulation scientist, I would say it's a product quality even also. But when we go realistic, so when this chromatogram can see the formulation managers, yes, they are the get confidence very high. And the manager always, I'm looking for an assistant that's not afraid of a little degradation when you have a such kind of product. So they are very confident on this. But in real scenario, when actual observation at analytical lab, yes, there are multiple impurities and multiple peaks, impurity A, B, C. I would just take an impurities and total impurities. So if this is the case as actual observation in an analytical lab, then what would be the CO expectation? I'm looking for an analytical expert who can support to provide the pathway to control the impurities. So now how the scenario changes from single peak to multiple peak and how many things gets involved when multiple impurities arises in chromatography. So the analytical part starts from here. So predicting the mechanism or pathways help designing the control strategy. I think we discussed little on this previous session also to minimize the potential chances of impurity formations. So now what is the role of analytical chemistry over here? To detect, identify and quantify these unwanted substances. First of all, we need to understand, again, we will back to that. Degradations, drug occur through the three principal pathways. And we need to understand these three principal pathways in very deep. Chemical degradation pathways, mechanical, I would say, sorry, physical degradations. We will not touch much on microbial because it's out of, I would say, subject for us. So chemical degradation pathways and physical degradations pathway. When we look at the chemical degradation pathway, there are multiple subpaths happens. We generally know on acid, base and peroxide and we will close this. No, there are multiple other pathways which we miss sometimes. So hydrolysis, we are always look at that, but we never look at the solvolysis. That is also happens in some of the molecules. Oxidations, we just explained in our previous slides. Simply, we always depends on oxygen and simple oxygen. Never we talk about the triplet state, singlet state, and superoxides. This is very important pathways. Photolysis, simply we will say the Q1B, 1.2 million lux hour happening, not happening. No, we have to go with the each wavelength kind of the sometime photolysis, and we need to understand. Polymerization, that is highly dependent on the molecules. Dehydration, if you look at the dehydration, it will impact to the physical state whenever dehydrate the molecules. Isomerizations, if you look, cis, trans, R, S, racemizations, optical isomerization and geometrical isomerization. So some of the cases we could not able to detect, but this isomerization, racemization is very important. We know the thalidomide case, it happens based on this optical isomerizations. When R and S and contradict potency or therapeutic activity. Decarboxylation and chemical incompatibilities. So these are the chemical degradation pathway. Generally, we evaluate during the any impurity determination detections. And once we detect the impurities, we will develop the mitigation strategy, which is we used to call it control strategy, which used to call it specification design, which we used to call it some process parameter or manufacturing system to be altered. Now, physical degradation pathway. Physical degradation, whenever it comes in the mind, we know ritonavir or carbamazepine. we knows this two case is very importantly how this is behaving. Polymorphism as a physical degradation pathway. Particle size, when we introduce 10 micron, sometime during the process it become 3 micron, and that have a higher charges and can happen a higher activation energy. Vaporization, during the physical process it happen, evaporation and temperatures. Efflorescence, hygroscopy and deliquescent, these are also physical degradations. So how the happen attrition, compression, shearing and impact on particle size impact, and how the ritonavir is happen from 1 to form 2. Even you grind also there is a change in polymorphisms, and sometimes you will expose, that is also a polymorphism. So there are many physical degradation pathway. This is also very important for a therapeutic activity of molecule. And microbiome, yes, it's a air, food, water and humans raw materials and finished product. So if we look at the case example, now audience now comes to you is very important. So this is the case example for you. Venlafaxine hydrochloride, we taken the case in the previous, where this is the ROS, I would say route of synthesis for this SM1, SM2, intermediate A, B, C. So you have a now questions to looking at this ROS. How many total isomer in this API you can observe? You note down how many isomers you have in this ROS. And you write down in the platform of chat, Dheeraj, ask them, so they can at least share their numbers or evaluation or answer. How many total process impurity from this ROS? And how many solvent impurities from this ROS? And total elemental impurities from this ROS, and total nitrosamine impurities from this ROS. If you can able to click this, I think my session will be meaningful to you. So let's we start this exercise. Let we list down the total isomer impurities, process impurities, solvent impurities, elemental impurities, and nitrosamine impurities. I will wait for a while to list down this all impurity, and then we'll go to the next slide. Dheeraj, can you just help me here to having yes or no for the next slide?
All right, everyone. So means, can you not stand behind me? That would be great. Thank you. Okay. So, everyone, I please request you to help and participate in this particular session. The session is not possible without your participation. And Shailesh, any poll or anything that you want to run selectively, that can be arranged as well.
Uh,
If
Gunit?
you have any poll about it. No,
Uh,
right? So you just want
no,
a
no,
response
I
from the
think yes.
audience.
Just I wanted to have any answer. And if it is any first answer will be right, you can have a gift also.
Okay, all right. Certainly, we can encourage that. And
Yes.
how much time we want to give to the audience?
I think 4 to 5 minutes is very much.
All right. Listen up, everyone. We have 5 minutes. I can put it as an announcement for you all. 5 minutes to solve. Can you tell me more about what are they going to work on in 5 minutes?
So they have to identify the isomers from this ROS, process impurities from this ROS, solvent impurities, elemental impurities, and nitrosamine impurities. How many numbers they have to write down?
All right. Okay, so I am putting this as an announcement for all of you once again. 5 minutes to solve, identify the impurities, total number of the impurities. If everyone understood it, can you please give thumbs up as an emoticon or put a message? Yeah, okay, everyone understood. All right, okay, so everyone is writing and typing, so we will give them 5 minutes so that we have everyone's answer, Shailesh.
Okay, Ganitya, thank you for supporting. So I think audience are ready.
Hi, audience. You have 3 more minutes left for identifying your answer and mentioning it in the chat box. Guys, 2 more minutes to go. All right, guys, last 1 minute to go. I can see there are many answers coming in.
Yeah, good, Dheeraj. So so they are not sleeping, it means.
Yeah, definitely.
Anyway, thank you audience for this support here.
Yeah. So what I'll do is, Seleji, after we receive in next 20 seconds maybe, we'll stop taking answers, and
Yes.
I will be showcasing. You know, mostly I can just give you a synopsis about what people are mentioning. Okay.
Yeah,
So
and let's have a token of something to them who has an first or second, if they are right.
yes, the
Yeah.
first person to mention. Okay, okay, all right. So we'll
Yeah.
stop taking the answers. The first person to mention an answer is Mr. Sanjeev Kumar. He's the director for process chemistry at ChemVeda, and his answer was 4 isomers.
Okay.
Then the second person, again, final API for Dr. Kaushik Singha. Again, he's the director for from Fresnes Kabi. Then we have Mr. Parminder Singh from Lotus Pharma, your organization. His
Yes.
answer is solvents 8 and elemental 1.
Okay.
Then again, Miss Shikha from Fresnes Kabi, she has given total isomers 2, process impurities 3, solvent impurities 8, elemental impurities 1, and nitrosamine 1.
Yes.
Then again, Dr. Sanjeev has answered solvents 8, then metals 2, then impurities 6. Yeah, that's for Dr. Sanjeev. And actually, people have given answers in multiple
Yes. Yes.
answers.
Yes. Yes. So let me take up Dheeraj now.
Perfect.
So
Yeah.
I think yes. So the exercise is a good exercise. I would say we can at least visualize there are many impurities on the ROS, but few impurities we knows we may not aware on this also, and few solvent out of solvent as a trace we also don't know. But if we list as a number, I think total isomer 2 as a S and R, I would say yes. I have not covered any OH related cis and trans. I think what is this doctor has given, but yes, it is also a part of that isomer. So it is always a debatable. Process impurities I consider 5, but not 5, 4 also, and sometimes it may be a 5 also, because venlafaxine is a base now here, so it may not be a considered as a process impurity for this. So it is always a, in any salt, base not considered as a impurity in chemical process, because it is already there in a way. So here SM1, 2, A and P, but yes, definitely other than that also there. Solvent impurity I covered here 6, but other than this 6 solvent, there are solvent out of solvent also would be a present. Elemental, yes, 1 impurities are here. That is a palladium. And nitrosamine, looking at the secondary and tertiary, we may not, yes, but yes, one of the nitrosamine can form over here if it is a breakable alkylation process happens. Yes, thank you audience for giving this support and evaluating this number. I think we can be live with this impurity evaluation process and identifying process. Yeah, thank you audience. Let me go to the next, and thank you Dheeraj for your support over here also. So now in the next, formic acid is a under solvent and impurity is organic impurities. So there are many case happens. What are the organic impurities and inorganic impurities? And impurities out of impurities are many. So there is a residual solvent. Formic acid considered as a organic impurity, it is considered as a organic solvent. And if looking at formic acid, I guess taken only one example over here to go little deep inside the any impurities we are missed sometime as a solvent and impurities. Which class? And that class is 3 in terms of the GTI, I would say genotox impurities. And that class, sorry, not genotox, class 3 residual solvent, that is not more than 5000 ppm. So if we know the formaldehyde, it's a non-mutagenic, not formic acid, formaldehyde. And that non-mutagenic formaldehyde also one of the GTA, and that is also go with the classification system in this assessment. And one of the methane sulfonic acid also go for the genotoxic impurity, and that is also go for the classification system. So look at the formaldehyde and methyl methane sulfonate, or out of that impurity isopropyl methane sulfonate, impurity of impurities, that is goes on class 1 and class 2 impurities, and that considered as a mandatory to monitor as a impurities under the classification if it is a potential. Now what are the commonly used impurity terms in pharmaceuticals we used? Audience, again I need your support here. So what are the commonly used impurity terms? Somebody we use says related substance, related impurities, impurities, degradants. There are many. Can you list down? Let we discuss on this part. So there is again I need 1 or 2 minutes, not much. So if any innovative or creative name also can come out, it is also good for us. We can ask ICH to implement that creative names.
Certainly, certainly. So, audience, you have 2 minutes time. Please send your responses in the chat box. I will read it out for Dr. Shailesh and we'll discuss accordingly. Yeah. Yeah, I can see there is one comment coming: degradation product related substance, uh, related substances, organic impurities, degradation products, related substances again. So there are three responses so far. Again, organic impurities, Deepak from Blue Cross has mentioned. There's one more response: organic impurity, inorganic impurity, solvent impurity. This is from Kamlesh, who is the EGM at Cohens. Again, I have a update from related substances, degradation products, organic impurities. Polymorphic impurities is one of the response from Dr. Sanjeev from Business Lobby. Residual solvents, organic, inorganic, residual, degradation, elemental, genotoxic, nitrosamine, process impurities.
So generally commonly used terms we knows.
There is one unique term which is side REI. I don't know. Mr. Surya Gopal Krishna has mentioned side REI.
Side reaction.
Okay, side
Side
reaction.
reaction products. Yeah.
Then we have enantiomeric impurities.
Enantiomeric impurities. Okay. So I think let me go to the next slide where we can have a multiple terms of impurities, and which as a drug substance stage, which as a drug product stage, we can classify differently. So several terms to describe the impurities. Impurities is penultimate intermediate, byproducts, transformation product, interaction product, related products, degradation product, foreign substances, toxic impurities, concomitant component, ordinary impurities, and organic volatile impurities. Yes, now ICH has to jump in this kind of the classification we generally term as a degradation product. No, it's not only the degradation. Wherever the any toxic substance, we have to write as a toxic impurities. Wherever any foreign substance related impurity, we should write as a foreign substance impurity. So the now time comes to the forum, we have to go beyond one step from the conventional and traditional nomenclature to the more deeper. Now the more in silico model is available. So we should go in this kind of terminology. But yes, we will wait ICH to update and implement kind of the things, and then we will implement in our regulatories, because we are on the generic process. Okay, audience. So let we have a unknown impurities now identifications process as a part of our now topic. So if any unknown impurities during our chromatography run we observe, so generally things comes the source. When we look at the source, yes, we have discussed very deep on the sources. We identified certain sources and we try to control. Still, we could not able to control that sources. Now the comes to the task to the analytical scientist. Now, how to make it a non? Because process and formulation scientists always wish to broaden the limit so they can get little leverage. Not I would say formulation scientists, but product quality shelf life also can be retained in the market. So if your isolation and characterizations, there are three different steps we generally use to follow. First is we used to do the extract out this compound. For extraction, generally we used to have a preparative separations or chromatographic separations. Preparative scale, if we go, then I think milligram to kilogram level scale also available in the markets. And if you look at the chromatography, there are multiple chromatographies we supposed to be used. HPLC, UHPLC, TLC. Sometimes TLC and flash chromatography is very useful, where only two isomers are there. HPTLC, helic chromatography nowadays. Gas chromatography generally less used preference for the isolation, but still capillary electrophoresis is also one of the good techniques to use. I would say organic and inorganic in a single chromatographs. It is very useful techniques for the biologics. Supercritical fluid chromatography for a isomeric and any two component kind of the process we can use. Detection, generally we use UV PDA for a spectrum identification, fluorescence for any pipi, and very concomitant I would say double bond system. ELSD, chemiluminescence nitrogen detector, sometimes we use where more nitrogenous compounds are there. Then corona CAD or charge aerosol detector for the kind of mass based percentage areas. And mass spectroscopy is well known nowadays, previously for bioanalytical and now for nitrosamines, and now isolation and characterizations. So wherever unknown impurities, generally we use this, follow these techniques for isolation identification part. And what are the characterization tools generally we used? When we have identified unknown impurities, we go through the separations. When we go through separation, then go through the small scale HPLC isolations, and then we will go with the MS and IR for a sufficient information on spectral part. And then followed by we will go with the NMR, mass, and IR, and collaborate these three techniques to determine certain structural informations. So in a larger way, if he says, because this flow chart is looks always a good, but generally when we start any identification, we are going with UV spectrum, then FTIR and NMRs if it is a pure form, and then mass, and mass MSN, I would say fragmentation pattern. And if it is a small biological, CD spectroscopy, circulum dichroism spectroscopy for any folding systems. So these are the characterization tool generally we use when we do the any identification of impurities. Now, what are the developing mitigation strategy once it identified the impurities? So we look at the gabapentin capsule. One of the case example is an anti-epileptic monograph reported one of the specified impurities related compound A as a lactam impurity. In long-term and real-time stability testing done for 36 months, an unknown impurity at RRT 10.5 was noticed, and it is beyond the identification threshold, that is 0.1%. Gabapentin, I think whosoever work, they knows how notorious and how critical in terms of impurity sensitivity. Stress testing of gabapentin during product development, conventional post degradation technique, that 10.5 was not detected. This impurity in its level in drug product could have critical impact on quality and safety, but it is only detecting during the stability, not in a stress. Since conventional degradation studies in that case may not happen, what we learn in a past slide, that is a realistic and idealistic. So this is not happens over here. Now, predicting unconventional pathway sometimes is very difficult. And many molecules in pharmaceuticals we have, which is a unconventional pathways for long-term stability studies happen. And it is very challenging for us. So here in oxidation condition, using autoclave was able to enhance impurity formation. So this is the structure of gabapentin. When we do force degradation study with not only thermal study here, if it use this terminology, thermal oxidation autoclave studies, where we have to be infuse some of the free radical oxygens over there. We have to provide constant temperature for certain period, that the energy will be constant, and that an impurity can be enriched. And then this impurity is enriched as unknown impurities by using prep HPLC. After the preparative HPLC, we followed through the WINOS. During identification, mass spectroscopy or NMR spectroscopy, we use it. Now in NMR, we general knows terminology of nuclear magnetic resonance spectroscopy. It's not only the single spectroscopy. It consists of 7 different techniques inside the NMRs. Where is 1H, 13C, TAP, noisy, apt, HMBC, HMQC, COSY, NOSY, Manning. And that is a structural characterization process. So after this process of thermal oxidation autoclave, because we used acidic degradation, alkaline degradation, oxidation degradation, photo degradation, we could not able to obtain that 10.5 RRT impurity. We extended some degradation with the metal induced introductions and I would say electron transfer oxidation process and free radical process could not happen. It only happened with the oxidation thermal autoclave process. And then after this impurity A, that lactam is becoming the dimer of impurity A. And this is very important information come through this stress study data. Because any impurity to identify it, very important to enrich that impurity. If we can able to enrich the impurity, then only you will identify impurity. Otherwise, very difficult for any process chemist or any analytical chemist to identify it as a structure for this impurity. So then impurity U as a dimer of impurity A identified in a gabapentin, and it could be leverages from 0.1% to 0.15% as a qualification threshold, being a more than 2 gram of MDD. And this can be a leverages to the specification, and we can able to extend the shelf life of this product. So this is the way of any impurities source identifying. After source identifying their realistic degradation pathway, and then isolation, and then detection and structure elucidation, and developing mitigation strategy through the proper specification control for the agency submission. Now, another case study, if you look at structural identification and risk assurance of unknown impurities in pramipexole, oral drug formulation. So pramipexole is a chiral molecule and is a non dopamine agonist used to treat various Parkinson diseases. Two unknown impurities in solid doses form at 30°, 40° observed. RT 8.7, one of that. And pramipexole having the composition, suppose mannitol, HPC, magnesium stearate, some of the silica, colloidal used. When pramipexole has mixed with mannitol, we can able to see this is the impurity of 0.38%, RT 0.7. I have taken only one impurity example. They are increasing two, but we will understand first impurity only. 0.38% is mannitol. If you look at the structure, there is a primary amine, secondary amine, some heterogenic structure also, and side chain ring also. So when they look at the chromatogram, unknown impurities increasing when we use with the mannitol, pramipexole mannitol, when we analyzed, it is increasing. So this impurity then later isolated characterized and identified, and it happens that it become the skip base, then amidori and glycosylation derivative. So we knows and we used to call it as a Maillard reactions in the industry as a broader term. Generally is a skip base and amidori rearrangement and glycosamine derivatives. So I think this case also will be useful for the primary amine and Maillard reaction. And first case is a dimerizations for a auto oxidation process. So then after identified the impurity, then determine the binding energy of this impurity. This is very important activity when any excipient related impurity is generating as a adduct. If we have some chemical information of binding energy and the dissociation energy, we can justify with the agency towards that. It is not going to be remain intact during the energy of the stomach. So this is also a docking and molecular dynamic simulation data that we can identify. And based on this data, we can leverage is the limit also and specification advantage can be kept. This is very unique way nowadays industry used it. But yes, many in silico model is required. And yes, risk appetizing also be required to filing stage. So the polymorphic form is a impurities. Ritonavir, yes, well-known case, form 1 and form 2. So the phase purity could have consequences for processing and solid state property and representing risk of product quality. So if we have a higher crushing or lower crushing energy provided to the monoclinic crystal habits, it maybe go to the orthorhombic crystal habits. So it happens very sensitive solid state molecules. So polymorphic form also very important to identify in some of the cases during the finished product manufacturing as a part of stability or maybe as a part of life cycle. Establishing patient-centric specification for drug substance and drug product impurities. So we used to call it term as a clinical relevant specification. This is nowadays using as a terminology set of criteria where acceptance range of drug products should be confirmed in order to deliver the therapeutic benefit. So process experience base, which is PEB specification, there are different pathways. Statistical limit estimated to cover most of the expected future result where tolerance interval, applying a mean plus or minus standard deviation of your analytical part, and using the range from minimum to maximum limit during the development. Here we are misguiding during the development sometime, and because of that is all specification and re-specification and re-revisions happens. So what process experience based approaches generally we use during specification? Risk that future batches will not meet criteria that were established based on the small number of development batches. So this happens or misguiding where we have a limited number of batches, limited lot we evaluated, and we set up some specification and then later said it become the consequences of reworking, rejection and reprocessing. Manufacturer with poor controls and more varied batch results likely attain more flexible operating space with wider limit. Batch failure related to inappropriately tight specification, sometimes we become very overconscious and over tight the limits, and that also become the failures in a systems and process. And widening the specification post authorization for a global product, we know how it's time consuming and how cost consuming. So generating limit for impurities with established toxicity data also sometimes help when we knows the anticipation of some impurities. So come to the end now. So evaluating the level of degradation product based on the risk benefit analysis is mandatory in industry. And if required, talks by design risk assessment methodology, which is TBD, generally not used in the ICH Q3A and Q3B. But yes, definitely has to be introduced. QTBD is a talks by design risk assessment methodology for any impurities or any adduct impurities for binding energies process based impurities. So this is not followed. Again, they come back to the same slide. Yes, we will be in the troubles with the same kind of the posters and news in the markets. So hoping this impurity identification and mitigation control and strategies helping to this audience to their routine work and routine way, and it will help to their own industries, I think respective industries, to make it more strengthen the area where we as an Indian state. So thank you, audience and team, and thank you Dheeraj, and thank you Gurneet.
All right. Thanks a lot for that. Can you hear me?
Yes, Dheeraj.
Thanks a lot for that session, sir. And I am sure that everyone understood about the identification of the impurities and developing the mitigation strategies. We'll quickly go to the questions which we have. The first question is from Dr. Kaushik Singha from Freshness Kavi. The question is it final API or RR or SS or RS or SR? So I think he's trying to ask about your following up question about your last concluding slide which you showed about.
Yeah, it's a racemic mixture. Yeah, this question is racemic mixture, R and S in an equal amount.
Correct.
And if it is an impurity, yes, definitely there is a 4 distereomeric, what he mentioned. He is also right. But if it is an impurity, but venlafaxine is a racemic mixture, RSSR.
Thank you so much for that. And we'll now move to the next question. It's can risk plans be different for different types of drugs or impurities?
Is very important, I would say. And yes, one need to follow the different type of drugs and impurities to be a plan with a different risk assessment protocols and process. We
Mhm.
are using one stick for the all molecules, not possible. We are using one stress study protocol for all molecule, not possible. We should look into the chemistry and we have to design the strategies on that. Yeah, I think she is very right on this case.
Thank you so much for that again. Okay, the next question is in case of unknown impurities, which was identified and limits that for NMT at 0.5% based on stability data, is toxicological data also required to be generated for this impurities?
If unknown impurity based on the MDD, if we consider as a point 0.1, generally drug substance 0.10 is a unknown impurity, and drug product 0.20 or based on the MDD is an unknown impurity. But yes, definitely if it is crossing qualification threshold, toxicological data is required to generate shiksha as per Q3A and Q3B class.
Okay, thank you so much for that. I believe we don't have any more questions. And again, thank you for asking. I'm sorry, and thank you for answering all the questions which were there. And you actually, went ahead of everything else, and you made sure that it is ending on time. And we value that very much.
Yeah, thank you, Dheeraj, for this. Yeah, and thank you, Gunit, for providing the opportunity to sharing my knowledge to this forum. And thank you, audience, for listening me after a lunch with very heavy way. But anyway, thanks a lot.